dear students, in today's video I will discuss about mainly the two topics. First is the mobility of the gut and secondly the mechanism of digestion. So let's start with the mobility of gut. The alimentary canal allows the movement of food forward along its length. You have already learned that it occurs through peristaltic movement. The alimentary canal or the gut shows the various type of movement. In the buccal cavity, there are two major functions. First is the mastication of food and then swallowing or deglutition. In the mouth, the food substances are broken into small pieces by the movement of the teeth. The tongue helps in the food to mix thoroughly with saliva and mucus. The mucus lubricates and adheres the masticated particle of the food into a bolus and pushes it forward the pharynx for deglutition. Deglutition. It is the process of swallowing of the bolus, that is the mass of soft food, into the esophagus from the buccal cavity through pharynx. The food is pushed back against the epiglottis. At the same time, epiglottis covers the glottis, due to which the esophageal opening becomes wider and the food enters into it easily. The bolus further passes down through the esophagus by a successive wave known as peristalsis as a reflex along with the constriction of the esophageal opening, which takes the food down into the stomach. The peristalsis is a series of muscular contraction that passes through the whole digestive tract. It pushes the food in the forward direction away from the mouth. The second topic here is the mechanism of digestion. In human being, the digestion of food starts in the buccal cavity itself. Then it continues till the anus of the large intestine. What happens in the oral cavity or the buccal cavity? The digestion starts in the oral cavity by hydrolytic action of the enzyme salivary amylase. The saliva secretes into the oral cavity contain electrolytes such as sodium, potassium ion, chloride ion and bicarbonate. And there are two enzymes which are salivary amylase and lysozyme. About 30% of the starch get hydrolyzed in the oral cavity by the action of salivary amylase at the optimum pH of 6.8. The starch is hence converted into maltose, which is a disaccharide. What is the role of lysozyme? Lysozyme acts as an antibacterial agent and kills the bacteria present in the food, thus prevents the infections. Next come the digestion in stomach. The food remain in the stomach for around 4 to 5 hours. Internal mucosa of the stomach contain gastric glands, which mainly comprises the three type of cell, namely the mucus cell or the neck cell that secretes mucus, the peptic cell or the chief cell secretes the proenzyme pepsinogen, and the parietal cell, which is also known as the oxyntic cell, secretes hydrochloric acid and the intrinsic factors. There is another enzyme known as gastric lipase. Small amount of lipases are also secreted by the gastric gland. Thus, in the stomach, the protein is broken down by the action of the pepsin at the optimum pH of 1.3 into proteases and peptones. Next is the digestion in small intestine. In the small intestine, the pancreatic enzymes like trypsinogen, chymotrypsinogen, procarboxypeptidases, pancreatic amylases, pancreatic lipases and pancreatic nucleases act on the food. The proteins, peptones and proteases, by the action of trypsin, chymotrypsin and carboxypeptidases, turns into dipeptides. The polysaccharide, that is the starch, which is not yet hydrolyzed by the mouth, is changed into disaccharide by the action of the pancreatic amylase. The nucleic acid present in the food turns into nucleotides and then further into nucleosides by nuclease enzyme released by the pancreas. Next is the digestion in small intestine. In the small intestine, the pancreatic enzymes like trypsinogen, chymotrypsinogen, procarboxypeptidases, pancreatic amylases, pancreatic lipases, and pancreatic nucleases act on the food. The proteins, peptones, and proteases, by the action of trypsin, chymotrypsin, and carboxypeptidases, turns into dipeptides. The polysaccharide, that is the starch, which is not yet hydrolyzed by the mouth, is changed into disaccharide by the action of the pancreatic amylase. The nucleic acid present in the food turns into nucleotides and then further into nucleosides by nuclease enzyme released by the pancreas. Liver. It is an important organ of the digestive system which releases bile. Bile contains bile pigments and bile salts. The bile pigments are bilirubin and biliverdin. The bile also contains some cholesterol and phospholipids. The role of bile is to break down the fat into diglyceride and monoglyceride. Remember one thing, bile does not contain any enzyme as the gastric juice has. So what does the bile do? It helps in the emulsification of the fat. Now what is emulsification? It is the breaking down of fat into very small micelles which are kept suspended in an aqueous medium. Next come the enzymes from the intestine. The mucosal epithelial of the intestine has goblet cells that release mucus. Along with the mucus, the intestinal juices are also released from the goblet cell. The intestinal juices together is called succus entericus that contain various enzymes like disaccharidases, dipeptidases, lipases, and nucleosidases. Thus, the digestion process is completed in the small intestine. Then the absorption of the nutrient of the food takes place by the help of the villi and the microvilli present in the wall of the small intestine. The undigested food further passes to the large intestine where the water and certain drugs are reabsorbed. Hence, the digestion process is completed.